Okay. Hi, everyone. Welcome to this month's webinar with Dr. Jean Bokelmann. So today, Dr. Jean Bokelmann, she will be giving a presentation on antimicrobial mechanisms of essential oils. She has been with NAHA for over 8 years, and she is a board-certified family physician, and she has been for over 35 years. So she has had a long-standing interest in integrative medicine and began formally teaching modalities to residents here in Idaho in 2004. In that same year, she actually helped open up the first endobiogeny clinic in the United States. And that was a collaborative effort of Time Laboratory and Idaho State University under the guidance of Jean Claude Lepraz. She actually has a book coming out this summer, and it is called Medicinal Herbs in Primary Care. And she is also going to be presenting at our conference this June, and her presentation topic is called Botanical Medicine for the Next Pandemic. So we are really excited for her presentation tonight, and I'm going to hand it over to Jean.
And so thanks for coming. And I really welcomed this opportunity to do a little research on this topic. Essential oils have fascinated me for many years, and my two favorite areas for essential oils are their applications in anxiety and in infectious disease. Anxiety is a problematic treatment area for, okay, we've got the, okay, I'm going to get the slides up here. There we go. So anxiety is a problematic treatment area for family physicians because a lot of the medications that are anti-anxiety are also pretty addictive. And so we try not to use them, and our options are limited. But the other facet of essential oils that also really interests me is infectious disease, because we also are challenged in medicine as far as treating infection and increasing antimicrobial resistance that's been happening. And it amazes me that one essential oil can be both can be all antibacterial, antifungal, and antiviral. And it's like, wow, how do they do that? So that my talk tonight is to explain how they do that, to satisfy my own curiosity, and hopefully to enlighten you and bring a more understanding, more depth of understanding on this topic. Okay, I hope, oh, sorry, I'm a little technologically challenged, so I'm probably clicking things I don't need to, but there we go. We have a little spinning picture here. So now I'll move on. This is where we're going. I'm going to talk about why essential oils are so valuable as an antimicrobial tool. And then I am going to go into the three types of microbes in particular, the bacteria, the fungi, and the viruses. And I'm going to talk about their structure, certain features about their structure, and how essential oils use these structures as their targets in having antimicrobial effects. So to start with, we have the problem with antibacterial resistance. We're looking at bacteria here. And over the past 10 years, what this slide shows you is that there are 10 different antibiotics mentioned here. And over 10 years, you can see the general trend for how they become increasingly resistant to antibiotics, especially if you look at those with the high spikes. Those are ciprofloxacin and levofloxacin, which are a fluoroquinolone class of antibiotics. And those are particularly prone to resistance. Sorry, I'm going to have to drink water frequently. I get a dry throat. But you can see that the other antibiotics, all these other classes of very strong antibiotics, are also having an increase in resistance with relation to the to various bacteria. Okay, and here's another example of antibacterial resistance or antibiotic resistance by bacteria. So this is something called beta-lactamase, and a certain category of antibiotics are called beta-lactams, and this includes the penicillins. Anything that ends with a cillin is a beta-lactam, and the cephalosporins. Maybe you've heard of Keflex. That's one of the most common cephalosporins we use. And because they are beta-lactams, a bacteria that can produce an enzyme called beta-lactamase can break apart the beta-lactam and render the antibiotic insufficient or ineffective. So you can see how the number of unique enzymes have increased exponentially in the past 30 years, pretty much 30 to 40 years. Oops, sorry. Now, what I want to show you just with this slide is kind of where we're going and the wow factor. You look at essential oils and you see these are the various constituents of various essential oils. And you can see that they have different targets that they have an effect on in a bacterial cell, bacterial organism, both in the cell wall and inside the cell. And I'm going to go into these in a little more detail. What's important, I think, about recognizing that you have, let's just say cinnamaldehyde, it's got a red arrow pointing to intracellular protein, and it also has another red arrow pointing to enzyme. It has more than one mechanism of action. And that's important because this is why we don't see the antibiotic resistance or the resistance to the essential oil the way we see it to antibiotics, because the essential oils not only have one constituent that has several mechanisms of action, but each essential oil has a number of constituents. So for example, thyme has thymol and carvacrol. And so if thymol has a few modes of action and carvacrol has other modes of action, it makes it really difficult for the bacteria to find its way out of this trap that the essential oil puts it in. Okay, so we're going to talk about these three microbes: bacteria, fungi, and viruses. And what I just want to do as an overview is point out that bacteria have differences that make them not uniformly susceptible to a certain essential oil. For example, let's say thyme and cinnamon. Let's take those two as an example. Not all bacteria are susceptible to thyme, and not all bacteria are susceptible to cinnamon. And there might be some overlap and also some uniqueness about the antibacterial mechanisms. And that relates to some of the features like gram-positive and gram-negative, which I'll explain in more detail, and anaerobic and aerobic. So some bacteria need oxygen to survive, and others, the anaerobic bacteria, prefer a really low oxygen environment. And then encapsulation versus non-encapsulation makes a difference as far as how the essential oil might work or not work on it. With fungi, they're broken down into whether it's a single-celled organism, such as what yeast is. Yeast is just a single cell of a fungus, and the hyphae or mycelium are the long branchy filamentous things that we think of when we look at mold. And And so those are basically the two categories of fungi. In viruses, we'll look at a little more in depth in viruses, and you have encapsulated and non-encapsulated viruses, and that makes a difference also as far as the targets that an essential oil might have on the virus. And RNA viruses and DNA viruses, they're genetic codes. Some viruses have a genetic code that's just made up of RNA, and some have a genetic code that's just made up of DNA. So we'll start with bacteria. Now, bacteria are prokaryotic, and what that means is that they don't have a membrane around their nucleus. So if you look at this picture here, that purple spaghetti in the middle, that is the DNA of the bacteria. And there is no circle around it. There's no membrane in this picture. And it's just kind of free floating in the middle there. And off to the side, you'll see something called a plasmid. And that's a little subset of DNA that also gets replicated and passed on to the progeny of the bacteria. And the plasmid is a really key part, plays a key role in bacterial resistance. This is the really versatile and adaptable part of the genome that is able to make differences that make it resistant to one antibiotic after another. And also in the bacteria, well, you'll see that it has the little hairy things on it. Those are flagellae and hairs, pilots, and those are important for movement. And those are made of protein. And then the ribosomes are the little dots in the center of the cytoplasm. That's the protein factory. That's what takes the DNA and turns it into protein, reads it. It reads it in a process called translation and converts it into protein. And it's the protein that is basically the life force of the bacteria, because it's the structure of the bacteria, and it's also all the enzymes that are necessary for the bacteria having its energy and its motion and everything else. So without the proteins, a bacteria would not survive. Okay, so I think I'll just lean forward on this. So another concept I want to convey at this point is cidal and static. And cidal means killing and static means staying. So when we talk about antibiotics, some antibiotics are bactericidal. They kill the bacteria, such as penicillin, by breaking up the cell wall, or in penicillin's case, the cell wall is malformed and it doesn't really connect. It kind of falls apart. With bacteriostatic, that is an antibiotic that just stops the protein production or stops the energy production, so that the bacteria just sits there. It doesn't explode necessarily or implode, but it just sits there, and eventually, by process of attrition or involution, it eventually does die, but it's not as overt. And so I'll go into these mechanisms with essential oils and how essential oils are both bacteriostatic and bactericidal. And that's another advantage to essential oils, is that they can function as both. So we'll start with the bactericidal effects, and this is really all about the membrane. And the cell membrane, I'll show you a picture of it in a little bit, and you'll see how, when you look at essential oils, we know that essential oils are hydrophobic, meaning they don't like water, and they are lipophilic, they like fat. And the cell membrane it's kind of like an Oreo cookie with the really fatty center. And the outer, the cookie part, the chocolate part of an Oreo cookie, that's the phosphate, the outer portion of the membrane, and then inside is the really fatty stuff. And the essential oils really love that fatty stuff inside, and they just have to get through that outer Oreo cookie chocolate part and get inside there. And that it can be sometimes tricky. It depends on the makeup of that outer aspect. But once they get in there, they just kind of take over in that area. And what they do, as this slide shows, is that they can disrupt the membrane. They can cause the membrane to expand. They can cause leakage through the membrane wall, and they can cause the membrane wall to just burst apart. Also, there are protein molecules within the membrane itself. And just by virtue of the essential oil presence in the membrane, they have an adverse effect on the proteins in there. And I'll talk, sorry, I'll talk more about that in a minute. And then the last thing is, the last thing that's important about bactericidal effects is that there is a synergism, so that one constituent may actually get into the membrane and do a lot of damage, but maybe it has trouble getting through that outer cookie part I'm talking about. And so there are other constituents that can help that get through. So for example, P-cymine increases the swelling of the cell wall of this B cereus cell wall, and it allows carvacrol to get through the wall and into the membrane. So this is the synergism of some of the constituents of an essential oil, and why they are just so intelligent without really trying to be. Periplasm is another part of the cell wall, because we've got the cell membrane, which has several layers. And I mean, we have the cell wall, and the cell membrane is one layer in the cell wall. The periplasm is another layer that can also have bactericidal effects from essential oils. So here's a picture of the lipid bilayer and the chemistry of it for those of you who just love chemistry. So if you look at the chemical formula here and you start at the left, you'll see that there is an attachment of several methyl groups, the CH3 methyl groups. And then as you move further to the right, there is the phosphate, the group there. And then as you move farther to the right, and then you see that the green hydrocarbon chain, that is the lipid part of it. Lipids are hydrocarbons, and they repel water. They're hydrophobic and lipophilic, and this is the place where the essential oils are at their best. But they have to get through that hydrophobic part first. And if you look at down at the bottom, the bilayer sheet, it shows that you have the phosphate and the methyl groups, let's say, the polar head group, on the top and the bottom of the membrane. And then, so that's why it's a bilayer. It's two layers, where the phosphate is on the outside, the hydrophilic part is on the outside, and the hydrophobic part is on the inside. So the essential oil just has to get through the hydrophilic part and into the hydrophobic part. It's a little confusing. Hydrophobic, lipophilic. Okay, so here's another picture, and I hope it's not confusing. What I'm trying to show, and I guess my arrow isn't able to translate through Zoom, so I'm just trying to describe it to you. If you look at number 1 there on the upper right of the picture, it shows that you've got the essential oil is increasing the membrane permeability. And what happens is that then molecules can flow through the membrane that normally are kept out for a reason. And then if you look into the upper left of this diagram, it points out that then you get leakage of the cytoplasmic materials. The materials from the inside leak outside, like nucleic acids and proteins. And when things leak out, it kind of shrivels up, and then the cell basically has what we call bacteriolysis. It just kind of falls apart or explodes. It implodes or explodes. And here's an actual photograph, a micron photograph of, it was a research done using citrus medica essential oil. And this was done on a bacteria and on Staph aureus. Staph aureus is the round things on, if you look at, you've got an upper and lower photograph there. And the upper photograph on the right side of that photograph are the Staph aureus. They're nice, plump, round bacteria. The things, the rod-shaped, the long square things on the left, that's not Staph aureus. So just disregard those and just look at the Staph aureus to the right of that slide. And this is before being exposed to the citrus medica essential oil. 4 hours later, they took a photograph showing the Staph aureus, and you can just see that, and it's Staph aureus across the whole slide in this photo. And you can just see how everything is smudged and broken apart and just looking very unhappy. So it shows how the citrus medica has lysed, has had bactericidal activity by virtue of its effect on the cell membrane. And here's just some examples of the essential oils and the effects that they have on the bacteria. And it's pretty much, if you look up and down this slide, it's all pretty much the same. So you've got Allium sativum inducing leakage in E. coli. You have Funiculum vulgare causing loss of membrane integrity in Shigella. You have cinnamon disrupting the cell membrane in E. coli and Staph aureus. So again, it's showing that this is a pretty common mechanism of action for essential oils. And I mentioned that you have the integral membrane proteins. That means proteins that are inside the membrane. And some of them go through and through. Some are attached on the outside and some are attached on the inside. But you can imagine how you get an essential oil in that lipid bilayer there, kind of pushing and moving things around. And it's going to distort these proteins. And if these proteins get distorted, then they don't work. And these proteins are very important. They're important for transporting. The ones that go through and through are transporting molecules. The ones that are on the inside are often part of an inner receptor, or what we call signal transduction, where it takes a signal from the outside and translates it to the inside without the molecule actually going through. Or proteins on the outside could be receptor proteins or other types of proteins. So anytime you get something in that lipid bilayer, it's going to mess with those integral membrane proteins. And that messes with the whole bacteria. And so here's an example of a membrane protein called the sodium-potassium pump. And that's something that's in throughout life. In all membranes, you have a sodium-potassium pump, and it pumps sodium out and potassium in. And it's important to have that in order to maintain a membrane potential in order for a lot of things to happen that I don't have time to go into, and probably not the knowledge either. And so this is another way that it's a very easy way for an essential oil to totally disrupt things. And when that membrane potential gets disrupted and it can't maintain that difference, again, the cell is likely to burst or shrink, the bacterial cell. So I promised I would talk a little bit about gram-positive and gram-negative, and I'm throwing in acid-fast bacilli as well. Acid-fast bacilli are hard to treat, and they are things like tuberculosis is the one that we struggle with most in medicine because it has a lot of drug resistance. So I wanted to talk about that a little bit as well. So gram-positive bacteria, this is kind of the description, and then I'm going to show you pictures because a picture is worth 1,000 words. But gram-positive bacteria have a thick peptidoglycan layer and no outer lipid membrane. Gram-negative bacteria have a thin peptidoglycan layer and an outer lipid membrane, as well as a main lipid membrane, as cell membrane. They also have a lipopolysaccharide in the outer membrane that restricts the diffusion of hydrophobic compounds. So you can see that this might be a problem for an essential oil to get through that. And it might create more of an obstacle for an essential oil to be effective against a gram-negative bacteria. And then the acid-fast bacteria have mycolic acid in the cell wall that makes them resistant to acid, so they won't take up an acid stain. That's why they're called that. But the mycolic acid also plays a role in some resistance to things trying to get in there. So here's gram-positive versus gram-negative, and what you can see is the peptidoglycan on the left is very thick, and that's the gram-positive, and on the right it's very thin. And then if you look at that bilipid membrane, you see on the left there's one bilipid membrane, and then the blobs kind of going across it are the proteins. And then on the right, you have a bilipid outer membrane for a gram-negative and a bilipid inner membrane, which in a way would make it more susceptible to the effects of essential oils. So if they can just get past that lipopolysaccharide layer, they can get down in there and really do some work. Okay, and then here's an acid-fast cell wall, and you can see the mycolic acid is up there at the top, the little bluish purple U-shaped things, and then the phospholipid bilayer is down at the bottom in yellow. And again, they all have this phospholipid bilayer that is where the money is when it comes to essential oils. And they just have to work with all this other stuff on the outside, and the different chemical constituents in the essential oils can have advantages and get through some of that. But we won't go into the details of that today. And here is a side-by-side of all three, just because it's a pretty picture. You can just see the differences between the gram-positive, the gram-negative, and the acid-fast. So here is an example of one size does not fit all. You have sandalwood and vetiver that inhibit gram-positive bacteria, but they don't inhibit gram-negatives. You have cinnamon, clove, pimento, thyme, oregano, and rosemary that do a pretty good job with both gram-positives and gram-negatives. So they have some of their constituents have little polar groups on them that help facilitate passage through those outer obstacles. And then you have tea tree that it works well for gram-positives, gram-negatives, and acid-fast bacilli. And so it really packs a punch. And here's just a huge slide that I'm not going to go over other than just to look at. This is more for your reference, that hopefully you can get a reference to. But what I want to show you is if you look at the right column, what I have is in red text are the gram-negative bacteria, or names of gram-negatives. In the black text are names of gram-positive bacteria. And in the brown, if you can see it, is the acid-fast bacteria. So if you look at cinnamon, you can see that it does a pretty good job with all three. In fact, all of these, if you just glance down, all of these plants, cinnamon, tea tree, oregano, rosemary, sage, and thyme, all do a pretty good job with both gram-negatives and gram-positives. And then you have the tea tree, the cinnamon, and the rosemary have activity against the acid-fast bacteria. So they are able to get through all of those obstacles posed by those bacteria. Now we want to talk about not just the different constituents and how they help with different types of bacteria, but also looking at the timing of harvest. I thought this was a really interesting study done that, and if you look at this, we're going to start with the upper graph, And we're going to go from left to right, and you see where it says VS, FS, and PFS. Well, that's the vegetative state. So when it was harvested in the vegetative state, and the essential oil was from that, and this essential oil is Oreganum compactum. And so the essential oil from that, and then the blue color versus red color versus green color is different strengths of, it's half strength, full strength, and double strength. And so no surprise, the double strength always seems to work better. And what they're measuring here is the optical density, which reflects DNA leakage. So they're looking at how this essential oil of Oreganum compactum produces DNA leakage through the membrane of two different bacteria. The upper one is E. coli, and then the bottom one is Bacillus subtilis. And E. coli is a gram negative, and Bacillus subtilis is a gram positive. And if you look at the top, you look at the highest bar, that highest green bar, that is in the flowering stage. FS is for flowering stage. PFS is for post-flowering stage. So it looks like the flowering stage works the best on E. coli. If you look at Bacillus subtilis, and you look at the green bars again, it looks like the vegetative state on the left and the post-flowering state on the right are more effective at disrupting the bacterial membrane and causing leakage than is the flowering state. And what you also need to be aware of is that the vegetative state is high in carvacrol, has both carvacrol and thymol. The flowering stage has mostly carvacrol, and the post-flowering stage has mostly thymol. So you're looking at what is more effective for these different bacteria. And the conclusion is that at the vegetative state, the Oreganum compactum at vegetative and flowering states, high in carvacrol, cause greater loss of membrane integrity for E. coli, whereas for Bacillus subtilis, it was more effective when it was high in thymol in the post-flowering stage. So time of harvest is very important, and you can see where studies done that don't take that into account might have inconsistent results. So now I'm going to move into the bacteriostatic effects, and these effects are due to effects on DNA regulation or DNA downregulation. And for example, thymol, carvacrol, eugenol, and benzoic acid have been shown to downregulate the expression of bacterial genes related to mostly to proteins, to motility, so the protein is flagellin, to adhesion to enterocytes, so various proteins, how they glom onto the intestinal cell lining, toxin secretion, toxins are also proteins, and quorum sensing, which I'm going to go into in a bit more detail here in a minute. And what I want to point out, I circled there in red the CH3, the methyl group, because DNA downregulation or upregulation has to do with methylation and methylation of the genetic material. And when you look at thymol and carvacrol, you see that they have three different methyl groups on them, on each of them. And so they may well be doing this through methylation of the genetic material. Moving down to the next paragraph, the interference with energy production pathways is another way that essential oils have been shown to cause bacteriostasis, just bring the bacteria to a halt. And so interfering with the TCA, that's the tricarboxylic acid cycle, that's the cycle that takes sugar and turns it into energy storage through ATP. And so interfering with that through interfering with enzymes, because there's a lot of enzymes involved in going from sugar into ATP, and also destruction of other enzymes related to that. The other thing that essential oils can do is oxygenate unsaturated fatty acids, causing some free radical damage, which is interesting because essential oils are by far and wide antioxidants. So it's interesting that they can selectively cause some oxidative damage. This is about quorum sensing, and quorum sensing is something bacteria do to make biofilms, and biofilms are another bane of antimicrobial management in medicine because they will form coatings on things and make it impossible for the antibiotic to get in through this biofilm. And if you think about quorum sensing, it's kind of like on the left here, you have single bacteria, the little yellow marbles at the bottom there. And then on the right, you have like a skyscraper of yellow marbles. They're all grouped together. And it's kind of like the country mouse and the city mouse type of thing. You've got on the left the bacteria that's kind of floating around on its own, but once you start grouping them together, so the one floating around on its own lives happily out in the country in his little grass hut. And but once they start getting together in mass quantities, they do this thing called quorum sensing. They realize that there's a bunch of them out there, and they change their metabolism, and they start building these concrete skyscrapers. And the concrete skyscrapers can't be penetrated by the antibiotics or broken down by the body and the immune system. And so biofilms are a problem, and they arise through quorum sensing. And quorum sensing is done by changing the bacteria changes its protein production and sends signals out there that cause this whole biofilm production. And this is kind of a busy slide that tries to explain that, but it explains how an essential oil can inhibit the peptide expression. That the peptide is what gets expressed when the bacterial cells realize they're being crowded. And then the peptide goes outside the cell wall, forms oligopeptides, which are just little tiny chunks of protein. So the essential oil can interfere with that. It can also interfere with the receptor of the oligopeptides, that little green blocky thing up there. And so it could interfere with that. Or it can interfere with the gene expression that happens inside from the oligopeptides actually getting in and affecting the genetic machinery that makes build concrete walls instead of their grass hut. And so basically, any interference with that causes the bacteria to just no longer be functional in that milieu, and it eventually dies. And down here at the bottom left, I point out that rose, geranium, lavender, clove, caraway, rosemary, oregano, tea tree, and thyme essential oils have been shown to be effective against biofilms. And here's an example of biofilm inhibition and comparing it, and this is kind of the same vegetative stage, flowering stage, post-flowering stage. And the other two clusters of bars, colored bars, are with two different antibiotics, erythromycin and chloramphenicol, and comparing them. And so with E. coli, the essential oil is not as effective as the antibiotic, but if you see, it's also more, if you're looking at the E. coli, you can still see that the vegetative and post-flowering stages are the most effective at preventing biofilm formation. And then the bottom, the Bacillus subtilis, if you look at that, you can see the big tall bar there is the post-flowering stage. And that did better than erythromycin or chloramphenicol. So post-flowering Oreganum compactum essential oil is better than antibiotics at inhibiting biofilm. That's just summarizing what I just said. And then we're looking at antibiotic resistance. And remember, I talked about beta-lactam, beta-lactamase, the enzyme that breaks apart a beta-lactam antibiotic. And here's a list of essential oils that can inhibit beta-lactamase, that can inhibit that enzyme, and therefore reduce antibiotic resistance to the beta-lactams. And here's another way that antibiotics are resistant. They have little pumps in their membranes called efflux pumps. If the antibiotic gets inside, the efflux pump just sucks it up and runs it right back out again. And so that's a problematic cause of antibacterial resistance, antibiotic resistance. So, so essential oils can inhibit these pumps. As I've already talked about, how they interfere with proteins. The pumps are, remember, proteins that are in the membrane. So rosemary, helichrysum, eucalyptus, C. ambroides, mamortica, punica, and then some constituents, alpha-terpenine, copanol, epicubanol, geraniol, and eucalyptol have all been shown in research to inhibit these pumps and therefore work to help antibiotics not become subject to resistance. I do want to point out some important things, though, that we assume if it's good for this, it's good for that, and that's not always true. So here's a rosemary essential oil plus ciprofloxacin against gram-negative bacteria displayed as favorable synergistic effect. So rosemary helped ciprofloxacin work against gram-negative bacteria, but rosemary with cipro against gram-positive bacteria was shown to have an antagonistic effect. So you can't always count on it working that way, and you really have to follow the research to know for sure that this particular essential oil and this particular antibiotic are going to work with this particular class of bacteria. Okay, I'm going to talk briefly about fungi. I'll try to limit it to 5 minutes, and then we'll get on to viruses. So the fungi they have more membranes, more of those bilipid membranes for essential oils to work with, or to work on, work against, mess up. But they also have a cell wall. So they have a nuclear membrane, and they also have, inside the cell, they have mitochondria. That's the energy factory. That's where ATP is made. And they have endoplasmic reticulum. That's where protein gets processed. And then they have still the cell wall. Now, you've got the cell on the bottom left there, and then up at the top right, you have a picture of mycelium with the hypha. The hypha is circled, and down in the bottom right is a close-up of a hypha. And if you just imagine that yeast cell just extending itself, it's not dividing and multiplying and making, or it's just extending itself. It's growing an arm here and an arm here, and that's how mycelium are developed from something that would otherwise be a yeast. So I mentioned the interior membranes that you have, and comparing a fungus to a bacteria, and you just see that there's a lot more complexity inside the fungus, and more complexity means more targets for essential oils. And the fungal cell wall, though, has the main feature is the chitin in it, which is a polysaccharide in the cell wall, and that can create a little bit of an obstacle to getting to that almighty cell membrane we're trying to get to. And again, the antifungal activities of essential oils are the cell membrane damage, which does everything that I've already talked about with bacteria. And then inside the cell, here's again this on the bottom left is a picture of a mitochondria where the ATP is made. And And on the bottom right is just kind of a diagram showing the outer membrane and the inner membrane. So mitochondria have lots of membranes to work with, and the inner membrane has all these proteins that can really be messed with an essential oil. And again, fungi also make biofilms, and essential oils can also disrupt biofilm formation. And one more thing unique to fungi is that it has ergosterol, which is the cholesterol of the fungus world. And it's in the membranes, and essential oils can decrease ergosterol biosynthesis and can also bind to ergosterol in the membrane and produce leakage. And here's just a very busy for future reference slide just showing different essential oils on the left, what their constituents are in the middle, and what fungi, what members of the fungal category are susceptible to those essential oils. Okay, so now I'm going to talk about fungus in the remaining 15 minutes. My voice holds out. So fungal envelopes are, as you can see in this picture, the envelope is it's a phospholipid bilayer. And so you can imagine that that's a great target for the essential oils. Some viruses don't have an envelope. Those are the non-envelope viruses. And so an essential oil can't really use that as a mechanism of action and has to do something else. If it doesn't have a capsule around it, that phospholipid bilayer, then all it has is what's called a nucleocapsid, which are just protein subunits that surround the genome. The genome is either RNA or DNA, depends. Different viruses have RNA as their genome, different viruses have DNA. And some viruses have just a single strand of RNA or a single strand of DNA, and some are double-stranded, just like we are double-stranded. And that can make a difference too, as far as action goes. So what a virus does is it's floating around out there, and then it attaches to the cell. So what we're seeing here is the virus that looks kind of like our good old coronavirus is stuck on the cell there, and there's usually a receptor that it attaches to. Then it gets engulfed into the cell somehow, and then it gets uncoated in the cell, and its genetic material gets into the nucleus of the host cell. So let's say this is a human cell, and it gets into the nucleus there, the blue part. And inside the nucleus, a couple things happen. One is that the genetic material can just get copied so that multiple copies of it are made for packaging and sending out. And the other part of it is that it can get transcribed into good old messenger RNA, mRNA, that we're familiar with now because of the vaccines for the coronavirus. Messenger RNAs leave their messengers because they go out of the nucleus into the cytoplasm, where they are then translated in the ribosomes into proteins. So the ribosomes read them and make a chain of amino acids that becomes the protein. And then these proteins can go to the Golgi apparatus, which is kind of like the Amazon shipping center, where they are packaged for shipping out or moving around to other parts of the cell. And then all of this gets put together. The virus gets wrapped up in its protein parts. The genetic material has been replicated. It is pinched off in the cell membrane, and it gets extruded and goes about to find another cell to infect. So now here's a long list, and I'm not going to, well, I could read it very quickly, but I think that the main point is that essential oils can effectively interfere with almost every step of that process that I just described to you. And I'll go into a few more details. But first, I want to talk about modern technology and in silico or in silico. I've never heard it pronounced, so I'm not quite sure, but it makes sense it's in silico because it's referring to silica, referring to computers, and or Silicon Valley, you know, that sort of thing. So in silico research is just computer research. They're doing molecular docking studies. They take a model of a protein, and then they take the ligand, the little thing that they're testing, and they put it together to see if they fit. And if they do, that's telling us that these molecules interact in a way that either enhances or inhibits the activity of the target. And there's a lot of research that's coming out because we don't have time to do things in the test tube the way we've normally done. They're doing a lot of molecular modeling for SARS-CoV-2 as far as what might work against this virus, against various proteins. And what they have found is that eugenol, menthol, carvacrol, garlic oil, and other essential oils were studied and were found to have docking scores that showed affinities with various parts of these essential oils with various parts of the SARS-CoV-2 virus, whether it's the spike protein, that little thing that sticks out, whether it's the main protease inside that is really important to the machinery inside the cell, whether it's the RNA-dependent RNA polymerase that allows replication of the genetic material, or whether it's the ACE2 receptors on the human cell that the spike protein attaches onto. So here's some pictures of that. And this is, so down on the bottom left, you can see where it says ACE2. That's on the human cell. That's the receptor. The spike protein, that little upside down flower there that's coming down from the coronavirus up top, attaches to that. So if anything, if you have an essential oil that can attach to ACE2, it will block the attachment of the spike protein there. So then the virus cannot attach to this human cell. So if short of that, there's another mechanism, which is the transmembrane protease. So this TMPRSS2 is an important protease in the human cell that has to cut that spike protein in order for it to be able to come down into the human cell. If that spike protein isn't cut, it's kind of still on its leash and it can't get down in there. So some essential oils can interfere with the transmembrane protease. And also some of them can just actually interfere with the spike protein directly. And then off to the right in this blue field is the main protease. That's the one that I said is inside of the virus that sort of manages all of the production of protein. And when that's inhibited, then the cell can't make appropriate proteins. So it's not able to cut the proteins apart in order to make the viral proteins if it's blocked. And so some essential oils in these molecular docking studies have shown that they will bind with that main protease and keep it from making other proteins, which is basically a bacteriostatic effect. And this is just a picture to show you RNA polymerase and what it does to unzip. This is regular RNA polymerase that is unzipping a DNA and reading off of one side of the DNA and making a messenger RNA from that. So it's just a cute picture. So here we have some of the results from the in silico molecular docking studies, and these are essential oil constituents that they looked at. And I just wanted to show you, without reading the whole slide, that some of them bind with main protease. That was the one in the blue field that I was talking about on the last slide. Some bind with other non-structural proteins, and some bind with RNA-dependent RNA polymerase. Some bind with the ACE2 receptor. Some bind with the spike proteins. Some bind with other parts of the spike proteins, and some bind with the RNA-dependent RNA polymerases. So here's just a slide showing a nice list of viruses that have been affected by various essential oils, and there's not nearly as much research in viruses. I think this is a great field for somebody to, I mean, we need a lot more work in this field. But what I want to point out is that the RNA viruses are in red, the DNA viruses are in black, and the enveloped viruses are underlined. And you can see that most of these viruses that have been shown to be impaired by essential oils are the envelope viruses, because they have that lipid bilayer on the outside. But that's not uniformly true. If you look at the eugenol and eugenyl acetate and carofilene are effective against the non-enveloped viruses. So they probably work at the protein level. And this is putting it all together. You've seen this slide before. Just to again, to reiterate that the different constituents can work together and they have multiple targets, and it makes it less likely for essential oils to develop resistance. I put this slide in just to remind us that human cells also have are chock full of lipid bilayer membranes, and therefore we need to be careful with essential oils. We can't use them indiscriminately. The difference is most human cells are not dividing and multiplying as quickly as bacteria are, so they're less susceptible than the bacteria or the viruses are to the essential oils. And that's our saving grace with essential oils. And finally, I just want to say that the antimicrobial effects are only part of the story. You can see here there's mention of the antiviral effect up there at 12 o'clock on this little clock, the antibacterial effect down at 6 o'clock, antifungal effect at 4 o'clock, I guess. But there's also neuroprotection, anti-inflammation, immunomodulation, tissue protection, anti-cancer effects. So essential oils are just amazing chemical mixes that can do so much. And when I talk about, I'm going to be presenting on the using essential oils for the next pandemic, or for the one we're in, actually. And I talk more about some of these other effects, the anti-inflammation and the immunomodulation, as just additional benefit to the whole body. So it's not just about getting rid of the microbes, it's about taking care of the whole body in the process. And essential oils are just amazing that they can do all of this. So stay tuned, and thank you very much for listening. And I think that I will stop sharing this and turn it back over to Michaela, and then I will answer any questions or address any comments.
Okay, so it looks like you did have a few questions in our chat. Carol asked, how were the oils exposed to the bacteria, by ingestion or inhalation?
Okay, so, well, that's a good question. And most of the studies that I am mentioning here are just all in the laboratory or in the computer with the in silico. So these are not clinical trials that were done, and we need clinical trials. But it's difficult to do. And so a clinical trial would be the inhalation or ingestion by a person or maybe an animal. But all of these were pretty much done in the test tube.
Okay, awesome. And then Melanie asked, how are the oils reaching the bacteria? Which I guess
Okay.
is kind of a little bit like the
Yeah,
last
it's kind of
question. Yeah.
funny. Yeah, and this
And
again, this is kind of a this is a question for the I think, well, for the things that I cited, we know this is just test tube stuff. But the question is, how effective is this in the body when you're inhaling? Where is that essential oil going? And we know when you inhale, these essential oils are just tiny little molecules. They can get through a cell wall. They can get into the bloodstream. They can get through the skin into the bloodstream, or you can ingest them and they can get in that way. I'm sure there are studies that have been done looking at what they, the pharmacokinetics of essential oils. That's what how the body processes an essential oil, how it absorbs it, and how it distributes it and how it excretes it. But this particular research just really, we don't know enough yet. And I think we need to know more. It's such a wide open field.
okay, perfect. And then Julie, she asks, so how does the immune system allow so many bacteria to build up to trigger quorum sensing in the first place? And then she said, does it start with antibiotic resistance or an illness out of control? Can a biofilm continue to exist in a person who appears healthy?
Well, a person who appears healthy isn't necessarily a healthy person, but yes, a healthy person shouldn't develop biofilms. And the typical place where we see biofilms, and we have the most trouble with them, are in parts of the body where the immune system doesn't work, meaning artificial things like somebody who's had a total hip replacement and has a metal ball in their hip now. That the immune system really doesn't work as well on a metal ball as it does on human tissue. And biofilms can work really well on total joint replacements. It is just a nightmare for orthopedic surgeons who do total joint replacements when they see that the joint is infected because of biofilms. So that's, yes, the immune system probably usually has some kind of compromise in order for that to happen.
Okay, wonderful. Does anyone else have any other questions that they would like to ask Dr. Bokelmann? We can unmute if anyone would like to ask a question.
Those were good questions. I I appreciate them, and I wish I knew more, but I wish we all knew more. I hope I can inspire somebody to go do research.
Okay. In the meantime, I guess while we're waiting to see if there are any more questions, I just wanted to thank Dr. Bokelmann for joining us today for her webinar. It will be uploaded onto the NAHA homepage, so you will be able to watch it again if you want to, and we could provide the slides as well in a PDF for you guys to look over again. Just wanted to remind you of our upcoming conference this June that she will also be speaking at. If you wanted to register for the conference, you can go to conference.naha.org. The ticket price is $250 for NAHA members and IFPA members. And if you are not a member, the conference price would be $300. So let's see. Yeah, okay. I don't see any more questions in the chat, so I think we can wrap it up. Is there anything else you would like to say, Dr. Bokelmann?
Well, I just appreciate the nice comments people have been making. That means a lot to me. So thank you.
Perfect. All right. Well, thank you for doing this for us. We will
My
log off
pleasure.
now.
I enjoyed
Yes.
learning.
Thank you.
Bye.